This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 336, recorded on May 8th, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Good afternoon, Vincent.
How are you, Dickson?
I'm very well, thanks. And yourself?
Excellent.
Good.
Excellent.
No one asks Vincent how you are, so I thought I'd take that upon myself to do it.
It's probably dangerous to do
well,
so.
It is. It
it's
has
currently,
been in the past.
It's currently 26 Celsius.
It's a gorgeous day out there. It's a little bit hazy.
Yep, but it's been a nice week. It's very warm.
Yeah.
The leaves are all out, and that's it. Now for the rest of the summer, we will complain about the hot weather.
And at this moment, the tide is running up the river.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How's southeastern Michigan?
It's 83 degrees here, which is 28, and we have nice cumulus clouds.
Cool.
Oh, nice.
Good weather is everywhere. Also joining us from north central Florida, Rich Condit.
Howdy, everybody.
You got 90? You have 90 down there?
No, 85.
Cool. Is that
29C. Yeah, this is one of these weird times of year where we got the same temperature as you guys do. That's, sometimes they're 40 degrees Fahrenheit different.
That's true.
But it's been gorgeous here in late April, early May. Really enjoying it before the summer hits.
And also joining us from western Massachusetts, Alan Dove.
Good to be here. It is hot and clear skies, and I'm so happy with that.
There you go.
How is hot? 90?
88 Fahrenheit, 31 Celsius.
Wow.
Oh.
Yeah.
You put the Wovle away?
Oh, the Wovle is folded up. It's stored in the now empty firewood rack in the garage. Yes.
Mount Snowplow is gone.
It's all gone.
It's all gone. The street sweeper came through.
Wow.
It is summertime here. I love it. I'll be sick of the heat by about August, but it'll take that long before I'm sick of the heat.
Right.
This episode is brought to you by ASM Conferences. And ASM has a little ampersand in front of it. I think that's part of the official name. It's a conference series, different, something new, offered by the American Society for Microbiology. It's designed to showcase the best science in a small meeting environment. And each person who goes to such meetings is either a speaker, a poster presenter, oral abstract presenter, or chef. All right, nobody laughed.
I'm waiting for
Dixon,
a further explanation.
Someone outside is looking
I was waiting for the rim shot.
Yes.
I did it. It doesn't,
That's for you. That's only for you, Alan. Okay. So basically, everyone there is presenting. There are no observers. And only 50 people are allowed.
Oh.
And you can apply or be invited. So this is good. This is like a mini Gordon conference. In-depth discussions and so forth. It's a nice little format. Scheduled discussions and social functions as well.
That sounds good.
Which means receptions and alcohol, etc.
Yes.
And they're held over a weekend to minimize travel. I don't know what that means.
Wait, but
you still have to travel.
get there.
I know. I think the text here is
Minimize travel costs.
maybe costs or minimize disruptions with your weekly schedule.
Right.
You know what they mean.
Yeah.
Anyway, you can propose a topic for 2016. The deadline is May 15th. This is the last time.
They're all held in the same place.
Well, Dixon,
To minimize travel.
To minimize travel, they're all held in New Zealand.
Right.
I shouldn't say that because people will all
That
be
would be
applying.
wonderful.
I don't actually know where they are held. They're probably at different places.
Oh.
But it actually doesn't say here, right? But I bet if you go to asm.org/conferences,
Sure.
you can get the information about that. And remember, the deadline is May 15th.
Yeah.
Tell them TWiV did not send you. I'm just kidding. A very, very important person in the field of science has passed away. Alexander Rich, 90 years old, and he was at MIT for many, many years. And he was there when I was there. I used to see him walking around with his little beret. And he did many things. Does anyone know offhand what Alex Rich did?
I associate him with the crystal structure of transfer RNA.
Transfer RNA, really? I associate him with zDNA.
Yeah, that's what I was going to
too.
say. zDNA.
And Dickson, do you associate him with anything?
I didn't know who he was.
You know what? He doesn't know who you are either.
He doesn't anymore, that's for sure.
He doesn't know who anyone is.
No, I knew some of those giants, but I didn't know him.
Okay. Anyway, 90 years old. Boy, that's
a really nice picture of him.
It's a great picture.
It's a good picture. He looks like he's from the Basque region of Spain.
He, if we could make it to 90, that would be cool.
Absolutely. Mm-hmm. I'm working on it.
He was born in Hartford, Connecticut. I didn't know that.
Oh.
Huh.
His father, Max, ran a dry cleaning business. Huh. Interesting. Anyway,
I like this last paragraph. Dr. Rich liked to recall the time he presented his collagen research at a scientific conference and ran into his old mentor, who was Linus Pauling. Dr. Pauling, he said, remarked that Dr. Rich had spent a long time in his lab without accomplishing much, but that he must have learned quite a bit.
Nice, really nice.
He says, I felt it was probably a fair assessment. That's what Rich said.
He's an honest guy.
That's pretty harsh.
Right,
That's what you say about me, isn't it, Rick? Isn't it, Vincent?
Not even a little bit. He doesn't
say,
talk about you like that at all, Alan.
He's diplomatic enough not to say that.
In fact, he doesn't talk about you at all. No.
I wouldn't say that to anyone, about anyone, no.
That was...
Only, I like to think that all of my people did a lot and learned a lot.
Yeah.
All right, we have some feedback, and I think, Alan, you should take that first one.
Yes. So Francois writes, Hi, TWiV team. This one is for Alan. In TWiV 335, he linked the European defiance for GMO rice to a supposed protectionism of French farmers. Yes, we've got our fair share of nutcases in Europe. GMO bad, vaccines bad, mobile phone antennas bad, gluten bad, blah, 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 etc. But here are my issues with Alan's comment. French rice production is around 80,000 tons per year to compare to 700 million tons worldwide. Right. At this low level of production, you're not even in the stats anymore.
Right.
On the 160,000 tons imported into France, around 4% comes from the U.S. and 80% from Pakistan, Cambodia, India, and Thailand. Let's be honest, U.S. farmers will not produce at lower cost than those guys. So no, French rice farmers are not worried about U.S. GMO rice for economic reasons. And then goes on about Europeans and GMOs. There's some reserve about health and GMOs in the general population, but the GMO mess really began with the quote-unquote substantial equivalence concept, the non-GMO labeling, and most of all, reports on Monsanto's seed police. Another problem is the socioeconomic cost and food security issues raised by allowing some few all-powerful private interests to own the rights to control the free growing of an essential food source. Remember, in continental Europe, not so long ago, all means of food production were owned by a few powerful interests, the nobility and the clergy, that were enforcing their own rules and influencing the government through financial contributions. Really love the show. Well, Francois, I've tacked in a couple of links for your light reading. One is a scholarly economic analysis of the European GMO ban from about 2003, and the other is an editorial that links to a World Bank report on the subject, and the World Bank actually came out and said essentially what I said. But perhaps, though, it was the context in which I said it was a bit misleading because rice was not the primary issue.
Right.
Right. Corn and soybeans are the primary issues, and I think if you pull up the stats on those, you'll find that the American contribution of them is considerably larger.
So the point is that there is trade protectionism.
There is trade protectionism. It's not so much about rice. Okay. Rice was a bystander, but the easy way to implement this was to ban GMOs. Because then you would ban all American crops effectively in certain categories, and that would serve as a protectionist barrier. And it wasn't just France; it was the whole EU that was this way.
Okay. Well,
so that's what I was getting at.
Check it out, Francois. Seems reasonable. All right, our other follow-up is from Jackson Kiaora TWiV, listening to TWiV 335 on my way to class at the University of Otago in Dunedin, New Zealand, where it's currently 14C and partly cloudy. I realized there is an easy extension to always have the rim shot for a joke on hand if you use Google Chrome, and he gives a link to that. I didn't make the extension. I just found it and was hoping it would be helpful.
But seriously.
Seriously. Thank you for the wonderful podcast, Jackson. Thank you, Jackson. I hope we don't overuse it.
But we will in the beginning,
We will.
I'm sure.
Yeah.
It depends. It's really for Alan, right?
Right.
Because none of us make any jokes.
Oh, I don't know about that. I don't use Chrome. Otherwise, I'd be able to deploy this
Well,
thing.
we don't do it on purpose. Let's put it that way.
Man.
You know, I used to always use Safari, and then the Google Docs got wonky, and it kept quitting. And so it doesn't have that problem in Chrome. So
Of course, it doesn't have that problem in Chrome.
Yeah. But I mean, I would click on
Suspect this is not an accident.
on a show note, and it would crash, so I didn't need that. And often in the middle of a TWiV where I have lots of tabs on Safari, it would get sluggish. So I switched to Chrome for at least for TWiV.
So genetically,
Firefox works fine, but it doesn't work for Google Voice. So when we do the podcast that way, then I have to use Firefox.
I wonder if the naysayers for gain-of-function studies in viruses would be equally opposed to gain-of-function studies for improving the nutritional value of a food item like rice.
Do you think that's a gain of function?
I know it's a gain-of-function.
Yeah, you're adding genes to it, right?
And you're gaining functions.
Well, you know, you're asking these individuals who will be unnamed to be reasonable, and they're
that's
not
right.
reasonable.
Well, that's right. I mean, that would ban the research on that altogether.
Yeah, right. I mean, these guys are just out of line. And the reason I'm mad is because all of us who signed on
Permanently
to scientistsforscience.org, is that what it is?
I was the second one to do that.
Well, we all got emails from Mark Lipsitch saying, I started a new site called, what is it? SafeScience.org.
That's right.
Practice safe science. Wear a condom.
Hit the rim shot. Thank you so much.
I'm really upset about this because they are just irrational. They do not listen to logic. I have made a number of arguments in print. They're totally ignored. They just go on.
I'm actually sorry I mentioned this.
With this view. I know it's boring to hear me, but we do have a paper, actually.
Oh, really?
This Rimshot plugin took a rapid downward turn. I just...
I know, I'm sorry.
One moment we're talking about browsers, and the next moment we're
There goes
all
the
angry.
highbrow show all out the window.
I don't know how they got started on that, but you guys got to understand. This bugs me. It sticks in my craw that one
Indeed
guy
it does.
is
Indeed it
messing
does.
with science for no good reason. Zero good reason. And I love science, okay? I have a passion for it. And if you guys love your science, you speak out against it. But not enough people are because we're all too busy.
You were around for Jeremy Rifkin, right?
Yeah, it was.
Well, it's the same situation.
A little bit.
Some of the
Maybe-
same objections.
And maybe you have to add Dr. Oz, so maybe there's two guys.
Oh, dear. Rim shot, please.
You know, the thing about Dr. Oz is that, forget it, never mind. He's not messing with science, right?
Although he thinks he is.
He's messing it up in many people, but he's not changing the ability to do science.
No.
And these other individuals are, and I just think this is inexcusable. There's no reason for it. We have a paper from Science Express. It is called Life-Threatening Influenza and Impaired Interferon Amplification in Human IRF7 Deficiency. There are a lot of authors on this paper.
There are 32 authors.
Sounds like a
just counted
physics
them.
paper.
But the first one, I have to say, Michael Ciancanelli.
Yeah.
He has two L's towards the end of his name, which I will
And it
remind
ends in a
you.
vowel. That's
It's like
good.
me. It's like me, right? Like Racaniello, right?
That's right.
The last author is Jean-Laurent Casanova from Rockefeller. And on this are some people from Mount Sinai. And Captain Picard is on it as well.
And not only is it in Science Express, it's in the hard copy of the April 24th issue of Science.
You know, Kathy, I'm glad you pointed that out because I brought that article in as I toured out of Science the morning it hit my breakfast
So
table.
I have this here, March 26th is the
And-
date of that's when I first saw this paper.
You guys are going to be amazed at the fact that this paper is already embedded in the Wikipedia description of IRF7.
Wow.
Yeah, these people are really onto it.
They're amazing.
So we have talked previously about trying to understand the genetic basis for more serious viral disease in certain individuals. And we did talk about, in the case of herpes simplex infections, a certain fraction of individuals get encephalitis. The virus goes into the brain where it's not supposed to, and kids get encephalitis. And Jean-Laurent Casanova got a number of kids who had herpes simplex encephalitis and did genome sequencing and found changes in the genes encoding parts of the toll-like receptor 3 pathway. And this is kind of the same idea, except here it's for serious influenza. Why do some people get really serious influenza, life-threatening illness?
Right.
Well, especially when they're otherwise healthy.
Exactly.
Right. So
to other
it's
viral
not,
infections.
you get a lot of people. Influenza typically hits the people who have immune problems, the old, and nothing personal.
Who do you think is old here?
Who's going to take
I was talking to
offense?
Dickson. And people who have some other chronic lung problem, and that's obviously a bad situation, but it's not as surprising as healthy kids. And that's really what they're looking at, is why do some seemingly healthy kids show up in the emergency room and go into the PICU and sometimes die from this disease.
What's interesting is that people with known primary immunodeficiencies, T, B cell immunity, they don't get serious flu. So it's clearly something else, as you say. So we don't understand it. So they did this study of a single child, a 7-year-old girl who was named P.
2.5.
What?
She was 2.5 when she got the virus.
She is now 7.
Yeah.
She was
7
2.5
now,
when
we
she was
performed
6.
whole exome sequencing in a 7-year-old girl, P.
Right.
P with a capital P. And I think that's cute.
Mm-hmm.
I think it stands for patient.
Yes.
It could.
Indeed.
It could, but who knows? These guys could have thought of something.
As I read this paper, I got really impressed with P, because she's been through a lot, and then they come back and they stick her with all kinds of stuff, and she gives them all kinds of blood and tissue, and she's really made a contribution. And I looked at the end at the acknowledgments, and the first line in the acknowledgments is a thank you to P and her parents.
Yes.
So I think it's good.
Well, I think part of the key here is developing relationships, and Casanova and all his collaborators, that's what they do, and it's important because you have to keep going back to the well.
So they got a case report here in the supplementary materials. Yeah. She was born in 2008. This disease that she had was in 2011. So I think she was about 2.5 at the time.
Right.
Mm-hmm.
And she got flu and got seriously ill. She was in the pediatric intensive care unit and was intubated and was in serious trouble, but eventually pulled through.
Yep. So her infection was with the 2009 H1N1 pandemic strain.
Hospitalized for 20 days.
Ooh.
Amazing.
So the fact that this kid actually, she and her parents were willing to come back to get additional tests done is very impressive.
Yeah.
So they did her genome sequence to do this study, and she was born to non-consanguineous parents of French descent. And the reason this is interesting is because in the herpes simplex study, which we did, some of the parents were consanguineous, and that really influenced the inheritance of these mutations.
So I want to just spend just a minute here on some of these techniques. The whole exome sequencing. Okay,
Right.
So there's only a fraction of the genome that is expressed as coding sequence, about 1%. And so if you make the assumption that any given disease is going to be because of a mutation in coding sequences, you can reduce the amount of sequencing that you have to do on someone to identify the problem by looking only at the coding sequences rather than the whole genome. You reduce it by a factor of, in theory, factor of 100. And you can buy a kit. In this particular case, they use Sure Select Human All Exon 50 Mega base kit from Agilent Technologies.
Did you say that again for the listening audience in Guam?
And
They did not pay us for that, right?
No. And
well, why not?
What this consists of?
Really?
What this consists of is 120 nucleotide biotinylated RNA sequences the individually that represent individually different coding sequences or exon sequences. That there's something like 40,000 of them or something like that. And so what you do is take genomic DNA from whoever it is you want to sequence, and you hybridize it to this RNA. And then since the RNA-DNA hybrids, by virtue of the biotin on the RNA, are now biotinylated, you can stick those to streptavidin beads and then paramagnetic beads, and then isolate the beads. So now you've got DNA sequences representing only the exons selected out of the whole genome, and then you just sequence that stuff.
Nice.
To get 16 reactions costs you $4,600. I can't believe. I was talking about this paper. Blows my mind. This is just, they do this now. I'll bet you within 10 years. 10 or 20 years, your genome sequence, or at least your exome sequence, is going to be a standard part of your medical record.
Here.
Yeah, we had our son's exome sequenced.
Oh, is that right?
Yeah. So, but
is that what 23andMe is?
No,
No?
not
Okay.
23andMe.
23andMe is more like a RIFLIP thing.
Yeah, it's just polymorphisms. No, we did it through the genetics department here to find out if he had any obvious mutations, but they charged his insurance company like $8,000 for that. So now I know that the kit is $4,700.
They did it twice.
But they only found 2 coding mutations in his genome, and they were irrelevant to his disease.
Did they sequence you guys too?
No, I haven't been sequenced. No. If it's $1,000, I'll do it. 47 is a bit steep.
So whole exome sequencing really came on the scene in 2010, and I associated Casanova with one of those first ones. And so, there's a whole exome sequencing-based discovery of human FAD deficiency, FADD.
I just wonder how much we miss by,
Yes, right. Because it misses all the possible non-coding problems. Okay, there could be microRNAs, long non-coding RNAs,
right?
And down somewhere in the future when whole genome sequencing gets even better, this will probably be obsolete.
Right.
Mm-hmm.
Well, in this study, so they sequenced a total of 22 people, right?
Yeah.
Mm-hmm.
Yeah.
And this one, they got a hit, which turns out to be very interesting, and that we're going to talk about. Whole exome sequencing in this case certainly gave interesting information.
But in cases where it doesn't, you have
in
to
cases where it
wonder,
doesn't, it certainly could be non-coding.
Yeah.
So they found
21 other people.
Yeah.
They found yes.
I didn't have time to look at this supplemental data, but did they explain, did they go and do the whole genome sequence because of her severe influenza, or was she just part of a group that they had whole genome sequence of, and then, they figured out, or she got the influenza infection?
Seems that the way they describe it in the main body is that they did it because she had severe flu. But
they did her and a bunch of other. They did a whole cohort of people who had severe flu.
Okay, that's what I didn't know. Was that all 22 of them?
Yeah.
Okay.
Yeah.
So
I gather they also have been putting together a database of whole exome sequences, because then they mentioned in the supplemental 37 people from non-consanguineous families in their database, and so they use that for some of their analysis as well.
Is that the Exome Aggregation Consortium?
It might be.
Didn't know there was such a consortium. Anyway, this whole exome sequencing of P yielded 2 mutations in the gene encoding IRF7. One at position 410 from phenylalanine to valine, and one at position 421 glutamine to a termination codon. And so P is heterozygous for each amino acid change and for each mutation, and the parents were each heterozygous for a single mutation. So the parents had one and the other had the other, and P got both. Old Mendelian inheritance.
Oh, yeah. I'm just thinking of Mendel and his grave.
Yes.
Isn't it wonderful?
It's wonderful.
It's just wonderful.
So IRF7 stands for, Dickson, interferon regulatory factor 7.
Indeed it does.
You always want to know what things stand for.
I actually went online and got the X-ray crystallography structure
Oh
of
my
the complex
gosh.
and looked at that for a
Oh
while.
my gosh, miracles do happen.
It does. That miracles did happen.
Well, this is how you knew that Wikipedia IRF7
That is how I
article
knew.
had been updated.
You got
probably.
the structure from Wikipedia, Dickson?
No, I got the reference from Wikipedia.
Really?
I got the structure from the
So
protein
how many
database.
beta strands are there in IRF7?
I didn't count them. There's so many. I don't do things like that, Vincent.
IRF7 is very important. In fact, when it was first discovered, the title of the paper included the statement, "a master regulator of interferon
It's a very interesting
production."
title. All Japanese authors, as I recall.
Yeah, exactly.
Yes.
What's wrong with that, Dickson?
Nothing. I just wanted you to know that I've read it. I downloaded it. I printed it. And I tried to struggle through the abbreviations. You know, there's someone that works in your laboratory, and I cornered her, and I read a paragraph. And there's about 3 words of English, and all the rest are TLR 137.
Yeah,
and
that's a problem.
It is a big problem because it's not English at this point. You know, it's
So IRF7 is produced in what's there. It's present in the cell, and upon sensing of innate sensing of
right.
foreign
It's
poly
latent.
things.
It's a latent.
I don't like that word.
I do.
That refers to herpes virus.
No, but it's a latent.
It's a protein sitting there, and
It's
then
an inducible protein.
Shut up, Dickson.
Hit the drum roll.
No, I'm not going to overuse it. You haven't said anything funny.
Everybody else
In
laughed.
response to sensing of foreign antigens and nucleic acids, IRF7 gets phosphorylated, and when it's phosphorylated, it goes into the nucleus and then turns on interferon synthesis. So it's amplifying, and it's also induced by interferons as well. So very important. So this young lady, 2 amino acid changes, and so they started to ask what's going on. So the way IRF7 works, it binds to promoters for the interferon genes and turns them on. So they set up a promoter assay, and in fact, these 2 proteins, well, one of course is shorter and the other has a single amino acid change, is not very good at these activating promoters.
And they're bad in different ways, right?
Mm-hmm.
So one of them doesn't get to the nucleus, and the other one gets to the nucleus but isn't phosphorylated. Is that right?
Right. So one of them is always in the nucleus and the other one doesn't get there. So the one with the amino acid change doesn't go in the nucleus. And the other one, where it's a shorter protein, is stuck in the nucleus, which is probably because the export sequence is messed up.
And of course, the parents they each have a wild type copy,
Yeah,
so they're
okay.
fine.
Yeah, they're okay.
And then the child inherited these 2 problems.
Yeah.
So she's got one that's defective because it doesn't go in the nucleus, and the other copy is defective because it's not getting phosphorylated. So her IRF7 is just non-functional.
And I must say that they do refer in this paper to mutant IRF7 proteins, for which they should really be ashamed.
How many
proteins-
authors?
Proteins are not mutant.
Mutation refers to nucleic acid.
But of course.
And
Well.
so, but you don't realize this until you write a textbook. I understand that. So the way IRF7 works is it dimerizes upon phosphorylation, and these proteins do dimerize, but they still don't work.
Right.
All right. Now, what if you take cells from this person and infect them with influenza virus? So they take peripheral blood mononuclear cells, and they infect them with influenza virus and look at innate immune responses. So what are they using for the control here? In healthy donors here, they do the same experiment. Great induction of type 1 and type 3 interferon genes, but not in P. P has a big problem turning on interferon genes.
Yeah.
And once this they have a negative control.
Yes.
UNC93B, is that I forget what that is.
So that's one of the genes in which he found, Casanova found mutations that predispose to herpes encephalitis, and it's a transport protein that helps get the TLRs from the ER into the endosome. And so he knew before that this was important for that. So yeah, that also has a defect in this as well.
And they're measuring the signaling in these cells with a microarray assay. So they're actually looking at the sort of global expression pattern of different genes, and they've got a heat map here of the top hits, and P is really dramatically different than the controls. It's impressive.
They looked in P's dendritic cells, which are known to be important producers of type 1 interferons as a consequence of having IRF7 in them. And they found they measure interferon production in these DCs in response to influenza virus infection. No interferon production, alpha production in these DCs. And one of the interferon-stimulated genes, which is important for influenza, MX1, wasn't induced in these cells. The mother produced interferon just like control cells in response to influenza virus. And they went on and quantified the induction of all the type 1 and type 3 genes by in these dendritic cells. And P has lots of deficiencies, for sure.
So there's along with my random readings on this, the IRF7 also is important for immune surveillance for leukemias. And so would people with these two kind of defects be more susceptible to... I mean, I know it's not an infection, but if the mutation would arise, then...
I don't know. How would IRF7 be involved in tumor surveillance?
Yeah, exactly. I would like to know that too. Because,
they didn't mention it here. No, I don't know.
The history of the protein has that as its as part of its rich.
So they immortalized some fibroblasts from P. So I just have to tell you, I should put this on my final exam for my class, which is next week, and I know nobody listens to TWiV. The exam is on Monday. They immortalize them with SV40, and I could say, okay, class, how does that work? So then I can do a lot of experiments on make them immortal and do experiments forever. IRF7, low expression even after IRF production, not expression. IRF protein production was diminished after interferon treatment. Higher titers of influenza A virus after infection of P's cells compared with healthy control. So they clearly have an issue with controlling influenza virus infection. But if they add exogenous interferon, it protects the cells from this increased replication. They also made pulmonary epithelial cells from induced stem cells.
This blew my mind too, technically.
So this is where my colleague Hans Schnuck, who's on the paper, Hans Willem Schnuck, he does this, and he takes patient cells and makes stem cells and then makes epithelial cells from them. It's really cool.
So the story that I made up for this, okay, you can correct me if I'm wrong, is that you want to get pulmonary epithelial cells from P in order to look at their interferon response, see what the defect looks like in those. But if you were to try and establish pulmonary epithelial cells from P, you'd have to do some sort of really gross bronchoscopy or something like that. So what you do is take dermal fibroblasts and reprogram them to be stem cells, and then redifferentiate those into epithelial cells.
Because we live in the future. Yes.
Yeah. And okay, you ready for this? Making the stem cells is also a kit.
Of course.
Yep. Right? They now do this with the kit that they uses a Sendai virus that has the fusion protein replaced with the genes that do the reprogramming to make stem cells. So this is kind of a hit and run. Because the Sendai virus goes in, expresses those proteins, and then can't complete its replication cycle. So eventually the Sendai goes away. The proteins have been delivered. They do their stunt. and the cells are reprogrammed, and there's no residue from the original infection like you would have if you did it with a lentivirus or something like that, and you have integrated copies. So it's a hit-and-run process of making these cells. Incredible.
And then you step into your pneumatic tube and slide on home.
So I had contacted Schnook, and I said, I want to make some of these because we do rhinovirus. The medium is so expensive, I couldn't do it. It was going to be many tens of thousands of dollars to grow a bunch of cells.
Wow.
So I couldn't do it.
Give it a few years.
Yeah. So anyway, they want to do this, of course, because they want to look in her lung cells, right?
Right.
Which is where flu is infecting, and ask, is this defect a problem? So they have these cells, and they can infect them with influenza virus. They can see that interferon expression or production is reduced.
So they can just make parts of this patient on demand.
They can make parts, and they see that. Well, what interesting finding is that the amount of influenza virus produced isn't different in the wild type and mutated genomes, but the number of infected cells appears to be about twice as many in P's pulmonary epithelial cells, which may be an issue. Maybe that's why you get serious disease, because you have many, many more cells that are initially infected. So that suggests that this contributed to P's serious influenza, disrupting innate immunity. I guess the reason is that the proteins. So why is it that it's messed up? The one protein doesn't get in the nucleus, and the other one is stuck there, so they can't do their normal participation. However, she does have a wild type copy of each one, right?
No, she's doubly heterozygous.
Right.
So she's got one chromosome's got one mutation, the other chromosome's got the other mutation.
Doubly? Right. No, she's heterozygous for each mutation.
She's compound heterozygous, so she inherited one mutated version from each parent.
Right.
So one copy is the F410V, and the other copy is the Q421X, and she has no wild type copies.
That's correct.
All right.
And that's what this transfection is about in, I forget which figure it is, where they do the double transfection with both of them to see whether there's any sort of complementation amongst them in terms of the transport. And they also, at the same time, do each of these mutations along with a wild-type copy to see if they're dominant negative, which they are not.
Right.
Okay, which explains why the parents don't have a problem.
Got it, right.
Did they call her P because of the Mendelian inheritance?
Yeah, maybe. Maybe.
That just hit me.
Sweet pea, actually.
I think they called her P because she's patient, because they call the controls C.
Okay.
That's just a simple way of thinking about it. I was a little annoyed that in a lot of places they don't explain what their abbreviations are in their figures and sometimes in the text. So we have to infer, but that's what I inferred about the C for control.
This has become so typical for these densely written papers. Like here's this sentence: each mutation was loss of function in reporter assays driven by interferon B, interferon A4, interferon A6 promoters, figure 1C. That simple sentence took me about 20 minutes to figure out, digging through all of the stuff and figuring out what the reporter assay was.
This is one of my complaints. I mean, these journals are messing up science publishing. Totally.
I totally agree with
I mean, this is a huge manuscript crammed into a little space. It's Science Express. I mean, it's crammed into a little space.
No, the other one looked the same, though.
It looks the same.
The journal article looks
and,
exactly the same.
You know, there's a lot of supplementary data which you have to go scraping for.
Yeah, you're right.
It's not presented well. I think they're screwing up publication. I'm sorry.
I would agree.
Yeah, they are, and it's a transitional. Hopefully, it's a transitional problem. That this is because so much more data is expected, but they don't have the space for it in the print edition that they're still obligated to produce, and so they're still bound to the print world, but they're also trying to, it's a real mess.
I hope you're right.
It'd be worse if they actually wrote out all these abbreviations, though, because then you'd have to read these very long.
But the other thing is, scientific papers have been like this for a very, very long time. We talked about this in the How to Read a Paper section, that this is not you don't sit down and read this like you would an article in the New Yorker. You know that you're going to have to unpack information that's compressed. It's just that traditionally, the figures were usually the part that all the data appeared in, and that was what you had to unpack, and the text had at least some room to explain what was going on.
So, Alan, dare I say this because it sounds self-serving, but isn't that why we're doing this podcast?
Yes.
Okay, we're deciphering these complex, densely packed articles and making them knowable.
Well, there's a multiple issue
I mean, you
here.
can make more fun of me if you want,
First
because
of all,
I'm the uneducated listener, but ultimately I ask the questions that listeners want to ask because this is tough.
Well, look, most listeners can't even access these papers, right?
No, that's right.
Because they're behind a paywall. And I don't think they would want to read it all. However, I'm thinking about the scientists who want to read this. You know, us. I just think it could be so much more nicely presented and written in a better way that's a little more scholarly than just bang, bang, fact after fact.
Sure.
And it's not the author's fault.
No,
it's
not
the
at all.
journals that are imposing this.
It's the journals, and this is absurd. We have to take control of our destiny.
There are a few
Follow me
things
to freedom.
they could have explained a few of their abbreviations in the figure legends and so forth. I still maintain that there was room for that.
So P's history is, I found interesting.
Mm-hmm.
So they say that, normal birth weight and gestation, perfect APGAR score. There was a rash suggestive of measles after the MMR vaccine at the age of a year. 7 months after this MMR vaccine, she had a new rash with fever evoking measles diagnosed by her pediatrician. 11 months later,
she got subsequent,
Yeah, she got new measles MMR vaccine without complications 11 months later. She did not receive vaccination against influenza virus at the time. She's had multiple episodes of bronchiolitis, one with hospitalization at the age of 18 months. And that made me think RSV or parainfluenza viruses. And sure enough, when you look down at her serology later on, she is seropositive for, she didn't have any trouble with any other vaccinations. Seropositive for diphtheria, tetanus, pertussis, polio, Haemophilus influenza B, hepatitis B, measles, mumps, rubella, streptococcus, meningitis. She's seropositive for the following viruses without vaccination: HHV6, CMV, VZV, RSV, adeno, human parainfluenza viruses 1, 2, and 3, and seronegative for EBV and HSV1. So she's had a lot of encounters with the virome, and except for this kind of suspicious thing with the MMR vaccine and some more than probably normal problems with bronchiolitis, has come through okay. And they say at some point in this manuscript that they're thinking that this IRF7 thing is pretty specific for influenza.
Yeah,
and this history pretty much supports that.
Yeah, they say it's probably redundant for most
infections.
But for some reason, flu, you get serious flu with these changes. Yeah.
By the way, just as a side note, APGAR was a pediatrician here at Columbia University.
Right.
Yep, she was.
Right. So this gifted musician.
This kid doesn't have any. She's not a sickly child.
No,
she's had a couple of kind of serious. She had the hospitalization and so forth before, but she's caught viruses before and gotten over them, and and flu just really did a number on her.
One of the things that I wondered about when Rich was just reading about the rashes, I hadn't seen that part, but I'd read the part about her having serum positive for VZV, but not having had the virus. And so apparently the rashes for measles and chickenpox look similar at the beginning, and then they develop some differences. So depending on...
you got to wonder whether the measles was diagnosed correctly.
Right.
Got to look for Koplik spots, right?
That's right, Kopliks.
Look at that.
I think this kind of study needs to be done with people who have vaccine-associated polio. because that happens at a rate of 1 in 1.5 million kids, so it's rare. And I'll bet they have some innate immunity
some problem.
change, and I'd just love to be able to get somebody to do that.
Well, I thought this paper was absolutely stunning. I really liked it. I love the genetics. Okay, the fact that all the Mendel stuff works. I love the technology with the what they did is just astonishing. The whole exome sequencing and all of the pluripotent stem cell generation, and the fact that everything makes sense throughout the paper. With the cells not supporting influenza. Not supporting virus replication and all of the IRF7 not activating the right promoters is just amazing.
So was one of their controls, because I didn't examine it as carefully as you guys, was one of the controls to restore the gene and get the cells to be refractory to the virus?
Yes, they did that in figure 4, where they add back wild type IRF7 to the patient.
Very good.
And in a few years, they'll restore the gene to the patient.
That's what I was thinking. That's exactly my
next
question.
we have to get past the.
There are some major hurdles to
that. Yeah.
Yes, you want to know that you're not going to make things worse.
Yep. All right, I have a couple of snippets. And snippets seems to be catching on. People are writing in and say, "Hey, here's a snippet for you."
Yeah.
Good. Andrew sent in a number of articles about an outbreak of avian influenza virus in the U.S. Some of you may have heard about this.
Yes.
I hadn't heard about it.
You didn't?
I'm right there with Dickson. I read what you send me, Vincent. Right?
So there have been 3 different avian influenza viruses detected. The main one is H5N2 that have been cropping up on chicken farms throughout the U.S. And this is a big problem because they have to kill all of these chickens. So there's a great page over at the U.S. Department of Agriculture, and I'll put a link to it. And they put the total number of birds affected. And this is kind of like Ebola SITREP, right? This is avian flu SITREP. 25 million birds affected so far. Of course, they all have to be killed because they don't want the infection to spread. Mississippi is a big one. Couple California, Washington, Idaho, all big bird growing states.
Midwest, Iowa, Minnesota, Wisconsin.
What about Frank Perdue's operation?
I don't know. It's not broken down by. Well, actually, it is broken down by company. Commercial.
Maryland? He has a big operation in Maryland.
So there are. It's interesting. There's some backyard flocks. There are some backyard ducks, commercial turkeys, commercial chickens, mixed poultry. So turkeys and chickens mainly. Maryland. He's got a big operation in Maryland. I don't see Maryland here on this list.
Yeah, so he dodged a bullet.
So far.
Yeah.
So the New York Times actually.
Well, he lays his own eggs.
You know,
wasn't that one of his ads?
Thank you.
I'm sorry, it's hard to find the button.
Yeah.
The New York Times ran an article on this, and of course that gets everyone's attention. And I must say, so this is by my friend Donald McNeil, and he's not really my friend, but I say that in a sarcastic way. He interviewed Peter Palazzi. He says, this is funny, it takes a lot of virus to... So the concern here is whether this is going to infect people or not.
Right.
And, it's not a high concern, I think. And the person from HHS... And Shushat, what does she say? It's low. It's a low human health risk, not zero, but low. And so he interviews Peter Palazzi about this. It takes a lot of virus to get infected with these avian strains, but not much of human. And Peter says that's why people who sleep under their chickens in markets in Asia get it, and we don't get it on Fifth Avenue.
Right.
Which I've heard him say before. And he says the people who are taking care of these chickens and culling them should wear BSL-3 suits with respirators and so forth.
It takes a tough virus to make a tender chicken.
Oh boy, that's good.
Hey.
So anyway, you have to kill all these chickens to try and stop the spread. We don't know, birds flying around move influenza viruses around, so that's probably how they got into the U.S. And hopefully it will stop soon because
came from Canada, just like the weather.
Is that right?
Yep, swept in from Canada. I'm going to blame the Canadians, yeah. McFadden's not here right now, but I'll blame him.
Canada, yes.
Right. I also got it this morning and yesterday. A few listeners sent in an article that was also in the Times and was just published in the New England Journal of Medicine. Jeff sent this to us.
My student from class just sent it, even though class has been over for a couple of weeks. He wanted post-class brownie points.
Yeah, one of my students last night, this is going to make it on the exam for sure because this combines persistent infections with Ebola.
Look at that.
It's going to make it. But they don't listen to TWiV, and they should. They should listen to everything. You never know when you're going to drop a hint. Anyway, Jeff had sent it. I'd love to hear your thoughts on this article. The article is Persistence of Ebolavirus in Ocular Fluid During Convalescence. So this was a physician who had been working in Sierra Leone, got infected, went to Emory for treatment, had serious disease, 12 days on mechanical ventilation, hemodialysis for 24 days. He had multi-organ system failure. I don't know how he lived. I thought when you have multi-organ system failure, it's like, wow. Anyway, he took a
good hospital.
Good hospital. Emory.
We have a machine for each failure.
Yes.
And then he slowly recovered. And on the day of discharge, they found Ebola RNA in semen by RT-PCR. And then sometime 10 weeks after the onset of his initial symptoms, he developed eye problems. And these got worse. And basically, he developed uveitis, inflammation of the, like the iris, the other parts of the eye, the choroid and so forth, near the front there, but not the cornea, just behind it. And so, they were treating him, and it got worse and worse. So they took a little bit of fluid out of the lumen, the vitreous humor, right? 170 microliters. You can put a needle through it, man. And
this makes me cringe.
Yes.
And it tested positive for Ebola by PCR. And they got infectious virus by culturing it. Now, they also swabbed the conjunctiva and it got tears, and they were negative. So they say probably he's not going to transmit it to anyone. So it seems to be in the vitreous humor, which is that big central cavity in the eye. And this is causing infection of the cells and giving him uveitis. And wow.
Yeah, wow. So they said they gave a number for the RT-PCR. They didn't give a titer or anything like that. And the number doesn't mean a whole lot, but they said sort of qualitatively that there was a lot of virus there.
A lot of virus, yeah. So this is interesting. Now, Jeff had said, does the vitreous humor make a particularly good or unique place for the virus to reside? Are there other bodily reservoirs that could present a problem if the patient were to become immune repressed? So this is great because in lecture number 17 of my course, which the lecture was on persistence, I have a slide called Reduced Immune Surveillance. And I say, cells and organs differ in their degree of immune defense. The central nervous system, the vitreous humor of the eye, and other places are devoid of initiators and effectors of the immune response. Right. And that's what this is. And you know what's another site that is immune privileged? The gonad. And that's probably why it persists in these areas. There's not much of an immune response.
So what's your answer to that then? How do you get rid of it from those places?
Well, what they did was worry about whether they should give steroids because that could make it worse or better.
Yeah.
But in the end, they did give steroids for one.
Yeah.
And then they also went back and forth about whether they should give a experimental antiviral.
Yeah.
And they gave that, but now we don't know whether that was what caused his recovery or not.
So he did recover.
Yeah, he's recovered, yeah.
Is virus negative now?
That's a good question. Is he now virus negative?
They don't say that they went back into his eye.
Viral culture sampled 2 weeks later was negative.
Oh, okay.
Anyway, so why it's persisting here and in semen is most likely because they're immune privileged and they're not clearing the virus that's getting in there.
Yeah.
Whether you could give a person antivirals and clear this is a good question. I asked our physician consultant, Dr. Daniel Griffin, and he said an antiviral will probably not make it into the vitreous humor. He said that's why, for example, for cytomegalovirus retinitis, you actually have to put devices into the eye to deliver antivirals. So that will be interesting to sort out.
There's even a worm that gets into the eye that gets all over the body first as a larva. The one that Toxocara canis and cat eye. And when the immune system really comes down hard on it all over, the ones that find their way into the eye, they just stay there. And then they cause a lot of eye problems.
Yeah, it's a good place for them to be, right?
Yep.
So apparently, I actually did an Ebola lecture, and one of the symptoms of Ebola infection during the main infection is eye issues. You can get conjunctival bleeding and so forth. So the virus, and they have a nice summary here. There are eye issues during convalescence and infection. They know this from before. Marburg does this as well. So this isn't brand new, but you wonder how many people have this. Both semen and eye fluids. How many people after recovery have virus in there?
Exactly.
Well, one of the things that they point out is that there's been few enough Ebola cases in the past, so that we don't have much of a grip on sequelae down the road. But with this, now we're going to get a much better idea.
Yeah, absolutely.
He had other problems, joint problems, and et cetera.
Yeah, you wonder about synovial fluid maybe is another privileged site, so perhaps you'd find it there too.
One thing that I found interesting from the New York Times article is that he's blue-eyed, and at one point his eye that was infected turned green, and then it turned back to blue. Usually when they turn color, it's permanent, so the fact that it changed back to blue was kind of remarkable too. And then it said in the New York Times article, I think, that he himself is going back to Africa to help evaluate some other post-Ebola survivors.
Well, now he's presumably immune, right?
Yeah.
So he's going to keep an eye on it.
Yeah.
I was just testing your reflexes.
It takes me a second to get up to I have a huge screen. I have Saul's old 30-inch screen, so it takes a long time to get across. The eye picture was on the left lower side, and I had to get all the way up and click on it. Anyway, this is quite interesting, and the article is very detailed.
Yeah. The other thing that was in the New York Times article was that the physician that did the sampling from his eye was wearing a mask and gloves, but no eye protection himself.
Oh.
He had a gown, gloves, and a mask, but no goggles.
I guess in a post-Ebola patient, you should have more protection.
Apparently.
Yeah, and then as soon as they figured out it was Ebola, people went rushing back to the room to disinfect it and so forth. Yeah.
So the question is, how many people have persistent virus in these sites? Eye, gonads, CNS, cartilage, etc. That should be interesting to sort out.
Well, my guess is that this guy is going to help sort that out.
Yeah, we hope so.
He received extraordinary care.
Yep.
The people who recovered spontaneously, how sick were most of those people before they actually got better?
Good point. I don't know.
What people who recovered spontaneously? What are you talking about?
Well, when you look at the number of cases and the number of deaths, it's not 100%.
Well, I don't know if spontaneous is the right word.
Well,
they did have medical care, right?
Not a lot of them. I mean,
How
they
do you
just
know
laid
that?
in bed.
Do you know the numbers?
Well, I'm asking the question.
I don't know.
The ones who got medical care did a lot better. But medical care was not as
it wasn't like this medical care.
Yeah, of course not. Of course not.
They didn't all get admitted to Emory.
So he had multiple organ failure and all kinds of other things. So he had a protracted. He would have died ordinarily.
Probably, yeah.
So, but people who recovered on their own is what I meant to say. They weren't as sick.
I would say people who recovered.
People who recovered with less help than he needed.
Yeah, I don't know. That's
People who
a good
didn't
question.
have multi-organ system failure,
exactly.
would they? What would they have?
These protracted. I didn't mean to say it that way. The fact that he was kept alive, so to speak, until he was able to fully recover, must have allowed this virus access to places that they ordinarily don't get to.
It seems like he had it. It seems like he probably had it worse than other people who survived.
Right. So
that could be part of it. But do you remember the guy in Africa who has he had virus in his semen?
That's true.
I don't know if he got the same kind of care, had the same course, but that's an interesting question.
Yeah.
By the way, this is a good segue into the Ebola sit-rep. There was a total of 18 confirmed cases of Ebola this week to the 3rd of May. Guinea and Sierra Leone each had 9 cases, and that's the lowest weekly total this year.
Right.
But, it's not gone yet.
So back to this story. So he has full-blown Ebola, right? And then he recovers, and then he develops the eye infection.
Yep.
So when did the virus get in the beginning, right? Was it residing there for a long time, and then it just happened to start an infection? The sequence of events is interesting.
Yep.
I mean, probably in a certain fraction of infections, the virus gets in the eye immediately because we know there are eye symptoms. So is it happening in most people, and then some of them have sequelae? Should we sequence his exome and see if he has a susceptibility gene for eye infections? Lots of interesting stuff. Gosh, infectious disease is just so good. But don't take it the wrong way, people.
No, and he's a physician, and he was over there taking extreme care, and he still caught it. So that tells you something about maybe he is a hypersusceptible person.
Maybe.
So that segues into the snippet that I see did not make it into the show notes that I sent
Go ahead.
Vincent. So a paper in Emerging Infectious Diseases is just now out ahead of press. It's a July 2015 issue. Ebolavirus stability on surfaces and in fluids in simulated outbreak environments. So this is open access. And they studied virus under what they call hospital conditions and under more outdoors Africa-like conditions. This is all done in Montana, I believe. I think, all of it's done in Hamilton. And so they have temperatures of 21 and 2 different humidities, 40% and 80%, or 27 and 2 different humidities, 40% and 80%. And they look at the stability of the virus on glass, plastic, and TWiV from the suits, and find that it persists a lot longer than people had thought. And they make various comparisons. In the moister, more humid environments, the virus survives better. They also looked at survival in water and in dried blood. Dried blood, it can survive for up to 5 days, and in liquid blood outside the body for up to 2 weeks. 11 days on TWiV, 8 days on plastic, 4 days on stainless steel. That was in the lab hospital-like conditions. And then in the hot, humid conditions, it was only 3 days on TWiV and less time on the other 2 materials.
Somehow I think DuPont is probably not going to advertise that as benefit of TWiV.
Right. But they also point out a couple of limitations. They use standard volumes of fairly purified virus. And so in real life, there'd be different volumes and different matrices that could influence the stability. They used 2 different things for their blood testing. They spiked some human blood with virus and looked at that. And they also used blood samples from non-human primates. And so the non-human primate blood samples might not be quite so indicative of what happens in people, as they mentioned. And that the conditions in the laboratory where they were doing these experiments are sterile, but out there in the wild, there'd be bacteria and chemicals and other stuff that would influence the viability.
Sunlight.
Yeah.
So in previous outbreaks, one of the risk factors is contaminated blankets in a home of someone. Right. So this makes sense that the virus can, retain infectivity for more than a day.
Right.
That's consistent with that. You're going to put that link in, right?
I'll do that, sure.
You sent it to me.
I'll do it now. Yep. I have that up.
This episode's also sponsored by ASM Microbe 2016. This is the new combined meeting of the American Society for Microbiology, the general meeting, and ICAC under one roof. Biggest gathering of microbiologists from across the globe. Premier event for the best microbial science in the world. The only meeting that explores microbiology all across the spectrum, a real transdisciplinary experience. This should be very interesting. I'm gonna go to that next year in Boston. I always liked ICAC and I like GM, and so together it should be interesting, although you don't know. We'll see. Anyway, all the great programming of both meetings, disciplinary sessions, profession of microbiology, expanded poster hall, networking, track-specific lounges, continuing education, over 200 sessions, 5,000 posters, 7 program tracks. Pretty unique opportunity for these multidisciplinary scientists to interact.
You going to read all the posters, Vincent?
Am I going to read them?
Yeah, there's only 5,000 of them.
No, I'm not going to
Can't
read
even
them.
walk in front of them
No.
at a fast pace.
Yeah, you go and you start talking to someone, and then the poster session's over, and you've seen one, But it's a big hall where they put all these posters.
Wow, 5,000.
5,000
Goodness.
posters, all that paper. You should someday
And
they're
work.
going to have LCD screens instead of your posters.
That might be right.
You put a little USB thing on the side.
Look at you.
Right? Look at me.
That would be perfect.
Anyway, the call for sessions is until May 19th. All right, so they want you to suggest things that they can do. And you can go to asm.org/microbe2016 to do that and find out more about the meeting. Let's do a little bit of email. I'll take this first one from Martin, who asks, why don't you write about this? Or no, why don't you talk about this?
Because it's a load of hooey.
Yeah, this is nonsense. 53,000 paralysis cases in India from polio vaccine in a year. This is just wrong. There is
No.
not. There's no more polio in India. What this is talking about is acute flaccid paralysis, AFP, which we talked about. There's a very high rate of AFP in India. There's also AFP in the U.S., not as high. But we don't know what the causative agent is. They absolutely do not have polio virus in them. It's something else. It could be Entero 68. It could be chemical or neurological. Who knows? But we've
All
actually
through the article, they call it non-polio
Well, that's
acute
the official-
flaccid paralysis.
Yeah.
Yes, that's the official name of it. Is that the WHO is obviously in the process of eliminating polio. They are very closely tracking AFP because that's the onset of polio
Right.
symptoms.
But this
So-
article is claiming that it's
No, this
polio.
article is claiming that they've just renamed the disease. No,
Yeah,
they've
no,
not
that's not.
renamed the disease. This is the background level of acute flaccid paralysis that you see in the absence of polio, and that's why it's being tracked so closely. And the fact that it's being tracked so closely, of course, means that they're finding a lot more cases that nobody ever would have noticed before because nobody kept
Polio's
track of
gone.
this
Yeah.
against the background of polio.
We talked about this a while ago, and there's a great article by a journalist there in India who corresponds with me a bit. I will put a link in the show notes where he talks about these 50,000 plus cases of AFP and the attempt to figure out what they are and so forth. And it's a really nice article. We'll put that in the show notes. A short one from Fernando. Yeah.
We're both doing the same thing. Use the do not link
We'll do
insertion
the
when
do
you
not
do
link.
it.
Fernando, hi TWiVers. Some sanity prevailed at the U.S. Patent and Trademark Office for a change. Main claims of podcast patent invalidated, right? So the EFF made a big push, and they got that bogus podcasting patent invalidated. Thank you. 2 strikes for podcast freedom. Okay, Kathy, you want to take Marcia's, please?
Sure. Marcia writes, hi, you guys. Parentheses, guys being generic and including the feminine gender also. And I have to say I grew up in the Midwest, and yeah, guys is all of us. No problem. I'm one of your occasional correspondents, having first found out about TWiV when XMRV was booted about as a possible cause. I'd like to share a few thoughts about CFS, parentheses, or ME, or CFS-ME, or SEID. I want to write a comprehensive account, but since I've got CFS and the limitations that come with it, I'll just have to do the best I can. Please let me gently correct you, I forget which of you, on terminology. Someone referred to the condition as chronic fatigue. There are many situations and illnesses that lead to chronic fatigue, which means you're tired a lot of the time. Chronic fatigue syndrome is something more specific. One definition of syndrome swiped off the internet is a set of signs and symptoms that appear together and characterize a disease or medical condition. AIDS is an example of a syndrome. Another example is heterotaxy syndrome, in which organs form on the wrong side of the body. A condition or illness is often referred to as a syndrome when the cause is not known, but there is a consistent collection of signs and symptoms by which it can be identified. Sometimes the syndrome is named after the person who first described the condition. Sometimes the use is humorous, such as white coat syndrome, when a patient's blood pressure goes up just because they're in a doctor's office. In the case of CFS, the choice of adjective was unfortunate because the word fatigue is so common. If I tell someone I have chronic fatigue syndrome, they often say, "Oh yeah, I get tired a lot too." Common thought used to be, and with many doctors still is, that CFS is a diagnosis of exclusion. However, clinicians who have seen many CFS patients, and the patients themselves, if they've studied extensively, can usually recognize someone with CFS by a particular set of symptoms. First and foremost, the ever-present overwhelming fatigue. I'm not talking, gee, I'd like to take a nap kind of fatigue. I'm talking the taking a 15-minute shower wears me out and I have to rest for an hour afterwards kind, and sleep doesn't help. One of the most characteristic symptoms is referred to as post-exertional malaise, or PEM. If someone with CFS exerts themselves, they might be able to approach a normal level of effort for a while, a few minutes, an hour, several hours, a day, but then will crash and require one or more recovery days. The difference in how a normal person recovers, even a deconditioned normal person, and how someone with CFS recovers can be shown objectively with 2 VO2 max exercise tests performed a day apart. The data from this test cannot be faked and is useful for legal reasons such as applying for disability benefits. My own theory for why this happens is that the body is not recycling ATP properly and recovery has to wait for more ATP to be built from scratch, but that's just a guess. Another common symptom is brain fog, similar to what cancer patients call chemo fog. Next, signs of past or present infection by Epstein-Barr virus and others in the herpes virus family. Many CFS patients report a sudden onset in which they came down with a viral illness and never fully recovered. Other pathogens besides herpes virus have also been proposed as a trigger, but that's the most common one. When I was first tested for pathogens, many years after I'd started telling my doctors I didn't feel well, I tested positive for Epstein-Barr virus, cytomegalovirus, Mycoplasma pneumoniae, and 2 or 3 others that escape me now. When I saw those results, it struck me that the problem wasn't any one of those pathogens, it was that my immune system wasn't working right. Dr. Nancy Klemas thinks that the illness currently known as CFS is primarily an immune system disorder and/or possibly a type of general dysregulation of the body's systems. When the Institute of Medicine undertook a review of the state of medicine vis-a-vis chronic fatigue syndrome, like many other patients, I was concerned that there were no CFS experts on the panel. However, in the long run, I think that distance from the field turned out to be beneficial because no one can accuse the final report as biased. The report, Beyond Myalgic Encephalomyelitis Chronic Fatigue Syndrome, Redefining an Illness, was released on February 10, 2015. The only part of the IOM report I really take issue with is their attempt to come up with a new name. I think we might as well stick with what we've got, even if it's not great, until we discover the actual cause of the illness. There have been continuous name wars amongst various parts of the patient community for so long, I don't think adding a new acronym into the mix is helpful. Okay, I'm fading, and this is probably long enough as it is. I'm attaching the IOM's 15-page clinician's guide, which explains the current state of understanding better than I can, in case any of you are interested. Thank you, as always, for your entertaining discussions. Regards, Marcia, in Salem, Massachusetts. P.S. Dr. Nancy Klemas is a delightful person, and she would probably be willing to be a guest on your show to talk about why herpes family viruses may be the culprit in CFS and other viral-type stuff. You can find her through the Institute of Neuroimmune Medicine at Nova Southeastern University in Miami, Florida. She's the founder and director of that institute. So, thank you to Marcia for that really thorough description of some of the aspects of CFS from a personal perspective.
I think sometimes we just say chronic fatigue to shorten it.
Right.
Mm-hmm. But we do understand that it's a syndrome. So
Right, like we say Ebola when we mean Ebolavirus.
Yeah, exactly.
Or Ebolavirus disease when we mean that.
Ebolavirus disease when we mean, yes.
However, we need to be clear that some people are sensitive to this.
Yes.
Yes.
Of course. Got it. Rich Condit, can you take the next one?
Johnny writes, this is our friend in Cambridge. didn't know if you'd seen this. I thought it might be one for the for TWiV references on Ebola. While more clinical, it seems intuitive and something that could easily be carried out. She sends a link at the bottom. Which was this?
Derivation and internal validation of the Ebola prediction score for risk stratification of patients with suspected disease.
Okay,
you might have Ebolavirus disease if. Yeah.
Yes. Right. So this is basically a study that clinicians can use to clinically diagnose Ebola. Right. Also, yesterday I had the good fortune and pleasure of meeting...
to diagnose risk of Ebola.
Risk, yes. Yeah. Also, yesterday I had the good fortune and pleasure of meeting Professor Stuart Firestein from Columbia's Department of Biological Sciences. He was a speaker at an annual symposium on learning at Harvard Medical School. I asked him if he knew you, and of course he did, and mentioned that he'd been a guest on TWiV. I'll look for that episode. I was delighted to learn that he'd been a guest. His discussion of ignorance was provocative and an interesting view on teaching the scientific method. Hope there is sun and blue skies where you are. It's 14C with mostly sun and what appears to be alto stratus clouds. Enjoy.
Interesting. All right. Thank you, Johnny. And we did a TWiV with Firestein. It's somewhere in there. Check it out. Alan, can you take the next one?
Okay. Nicola writes, Dear TWiVome, I was going to start this email by saying, Arg, the TWiVome has to stop uttering falsehoods about CFS. But I quickly realized that when you do, it gives us patients a chance to write in with corrections. And where else can we get the attention of thousands of scientists and scientists-to-be whose curiosity might be sparked? Rich is mistaken about the history of the 37 kilodalton RNase L discovery that he mentioned at TWiV 331. It had no connection with XMRV. This discovery was made in the mid-90s in the lab of Robert Suhadolnik at Temple University, who published several papers on the upregulation of the 2,5-A synthase RNase L pathway in CFS, and gives a reference. And then Rich is going to have a reference that he was thinking of. Do you want to do that now?
Sure.
Okay.
So I forget exactly what I said, but what I had was a vague recollection of a link between an association between an RNase L mutation and XMRV. And frankly, the reason I remember this is because Bob Silverman came to UF and gave a seminar long before any of this was published, when he was talking about their research trying to trace or pin down a connection between an RNase L, a particular RNase L variant, R462Q, and prostate cancer. And they were studying this in a prostate cancer cell line. And I remember him saying at the time that they were becoming suspicious that there may be a retrovirus associated with this. And the idea was that since RNase L is an antiviral thing, that maybe a mutation in RNase L would make a patient more susceptible to a virus when the virus is really the culprit in the prostate cancer. And that's what gave rise to the original publication that looked at the prostate cancer cell line and discovered XMRV. Okay, so that's the association between RNase L and XMRV. What I wasn't aware of, that Nicola is writing about here, is that there was a much earlier association made between RNase L and chronic fatigue syndrome, where some CFS patients have some sort of smaller variant of RNase L that apparently is a proteolytic breakdown product. The RNase L is normally 80 kilodaltons, and this thing is 37K, and it's a proteolytic process, and so the proteolytic product. And this had been studied for some time, but importantly, it's that association between RNase L and chronic fatigue syndrome, and the later association between XMRV and RNase L that made some people say, aha, so RNase L equals CFS, RNase L equals XMRV, therefore XMRV equals CFS. Right. And that's where that connection came from, and that took off on the CFX XMRV thing. So it's nice to have that additional bit of information to complete the story.
Right. And continuing the letter, during the XMRV era, this earlier research was said to be in support of the XMRV work. There was an RNase L connection with prostate cancer, as Vincent mentioned. Anyway, the point I want to make is that the work on the 37 kilodalton RNase L and CFS is an earlier and independent discovery and not tainted by the XMRV work. Right. So the RNase L discovery, people, that kind of led people to the whole XMRV digression, but the XMRV digression is not does not color the earlier discoveries.
Yeah, that's
about
one
RNase
of the
L.
points that Nicola is making. Right.
Yes. So at one time, patients could be tested for this enzyme, and maybe still can be, and there's a medication, Ampligen, a synthetic double-stranded RNA that inhibits the pathway. Not all patients have this 37 kilodalton RNase L, so it's not a biomarker for CFS, but I have been told by several CFS doctors that it may be a marker for who would benefit from Ampligen therapy. There are doctors out there who do things for their CFS patients, although there are far too few of them. I've spoken to patients who have been greatly helped by this drug. I say this not as medical advice to anyone, but only to speak to the importance of this research. Dr. Suhadolnik's page at Temple describes his research in CFS, and she gives a link. Jenny mentioned in her letter, read on TWiV 330, some research on abnormal response to exercise. This work goes back to the early 90s, but progress has been very slow, and I can only assume that's due to lack of funding. I was fortunate to have had exercise testing performed in 1995 by a CFS doctor and to have it reviewed by an exercise physiologist, which taught me a lot about my disability and how to manage it. This kind of testing really needs to be more widely available, but I don't think it will be without more research. Ampligen has never been approved, in part because of the studies required for approval are costly. I started listening to TWiV to get a perspective on XMRV from scientists who didn't have a dog in the fight. I think you all did a great job covering that. I have to say, I did sometimes groan at your lack of knowledge about CFS, but then I wasn't listening to learn about CFS, I was listening to learn about the virology. At some point, I went back and listened to all the TWiVs from episode 1. Dickson's way of talking about science and his storytelling skills in the early TWiVs led me to start TWiP from episode 1 also, even though before that I thought worms were just yucky.
They still are, by the way.
Now I'm a faithful listener. Not all worms. Now I'm a faithful listener of all the Twix podcasts. They brought science back into my life after a budding career was cut short by CFS when I was very young. Many thanks, Nicola. P.S. No email to TWiV would be complete without the weather. Here in Connecticut, it's currently 39 Fahrenheit. That's 4C and cloudy. Oh, so this is just from a couple of weeks ago. We're experiencing frost before morning. They're expecting frost before morning.
It's not too long ago, actually.
Well, we've learned a lot about CFS, too.
Oh,
Yes.
Yeah.
Mm-hmm.
All right, Dickson, can you take...
I'll try. Alenia writes, "Greetings, TWiV, from Toronto, Canada, where after a beautiful weekend of 20 degrees C, we're experiencing rain, 4 degrees C, and 50 kilometer per hour winds. I have a sweater, coffee, and 2 dogs who are keeping me warm, though. I was the listener who wrote you about the WHO Ebola sitrep. And they list a reference. See, doesn't it make sense? Doesn't it make you feel cool to say sitrep? I just watched the Dr. Fauci ASM talk that Vincent picked a few episodes ago, and it reminded me to thank you for your continued updates on the Ebola outbreak in West Africa. I'm sure I sounded neutral in my first letter, but I was experiencing continuous outrage through the beginning of this year about how the rest of the world was dealing with the outbreak. MSF was screaming for help months before anybody started listening to them. In early September, several months into the outbreak, I remember reading Dr. Joan Liu's presentation to the United Nations, and she gives a reference to that too. And having to take a walk, I was so angry. When the outbreak was at its peak, most of the media were focused on stuff that shouldn't have mattered, Ebola spreading to North America or Europe, when the real stories were that people in Liberia, Guinea, and Sierra Leone were getting sick and sometimes dying so fast that we couldn't even count them, and that the world utterly failed in its response, its responsibility, and even its self-interest. As the saying goes, the price of freedom is eternal vigilance. So thanks again for keeping a level head throughout your coverage and for continuing to talk about the outbreak and the people and organizations who are fighting it. I feel like there are important lessons here and that we as a society should learn, and keeping the discussion going is an important part of all that. All the best.
All right. Thank you. One more. This is from John. TWiV team. Listening to older episodes, I listened to episode 252 and heard the comments about driving in electric cars and Alan bending over backwards to excuse driving a huge engine gas eater type car as okay. If you believe in science and actually care about the planet you are leaving to your kids, then driving this type of car or a truck when not needed is showing utter contempt for science and climate change. I know that Vince has said that his family was not that important to him, working all the time and not spending time with them, so it is almost not surprising that he would not care about what he is leaving for them.
I'm glad I'm not reading this letter.
Why?
Nothing.
I wouldn't blame you. Usually I'm behind what Alan and Rich and Kathy have to say, but recently Vince has seemed to be upset that Lipkin discovered MERS in bats, was upset that something was not to his liking in another story, and was adamant that he would prove it by starting to study it, and seemed to be dead set to prove it regardless of where the actual facts led. I do feel sorry for his family, and I'm really sorry that his son may have CFS, and I really mean that. I'm really sad that a show I have really enjoyed has seemed to go downhill so much.
See ya.
Sorry, goodbye. Most of what you said isn't right, John.
No, I don't recall bending over backward to justify huge engines. By the way, my cars both have small 4-cylinder, normally aspirated engines, and I consider that important in buying one. But yeah, this is not. You're obviously listening to the episode with expecting what you're going to hear, and it's not what we're saying.
So what I was upset about Lipkin is that he found 190 nucleotides of MERS in bats, claimed that was the source, and it wasn't. It's probably camels, and so I don't know why you're criticizing me for that. I mean, and that I want to prove things regardless of where the facts led. That's totally not my science. So you got it wrong, John. I'm sorry you don't like the show. I really. What did he say? I lost it.
Really sad that a show I've really enjoyed has seemed to go downhill so much.
Sorry. I guess that's why our listenership just keeps going up.
That's right.
All right. So all the next bunch from Anthony, we have to do with the chicken outbreak.
Yes.
Which we've already talked about. And so I guess we could now stop right here. I'll stay end email.
That'll be good.
And then we'll go on to listener picks of the week. And now I have to scroll for a half hour before I get there.
While you're doing that, I looked up the Stuart Firestein was between episode 212 and 213. If you go in the TWiV archives,
special, right? Yeah.
It was a special. But you can just search on the site for Firestein or look between 212 and 213.
Of course, if you just Google TWiV Firestein, you'll find it too.
Yep.
Because we have good Google foo. Okay, picks of the week. Alan Dove, what do you have this week?
I have a time-lapse movie of a Winogradsky panel, which is basically a glass box full of dirt. So this, I realize on its face, a time-lapse movie of a glass box full of dirt does not sound very interesting, but I suggest you watch it and then reach a decision. So it, and the blogger who posted this explains what's going on, that you're watching the succession of different bacterial communities in this mud between the glass plates, and that's of course the point of a Winogradsky panel. You can Google that and read the history of Winogradsky columns and such. And it's just really neat to watch this time lapse, and you see the different colors of bacteria, and I assume algae. There's a lot of green stuff in there, changing and taking over different parts, and little colonies get established and grow and die. And I think this sort of thing is cool.
I wonder what it smells like. Too bad they can't put the smell in there.
Someday. So this is David Esteban, who has written quite a few times, a pox virus guy up at Vassar. I think I remember him doing this a while ago. He just published this, but he actually did this a while ago. I think he tweeted about it. Yeah, it was pretty cool. Dickson Despommier, what do you have for us?
Well, I have sort of a compilation from the National Geographic of landscape photographs that are absolutely soothing and astounding and startling and all the above. So when you have nothing else to do, just put a couple of those up and full screen them, and you can crawl into parts of the world that you may never get to.
Well, I guess I'll never look at them. Dickson, are your pictures in here?
I'm not that good a photographer.
Dickson, do any of them have pictures of water that Vincent might enjoy?
Or angiosperms?
Yeah, there's some underwater pictures even.
Yeah, he might get a kick out of some of these. I think you'd get a
I don't
kick
have
out of all of them.
I don't have free time because I don't like my family, so I don't have free time.
Vincent almost became a photographer before he became
No, I didn't
a
even come
virologist.
close to become.
No, but you had aspirations.
I did, but it was misguided.
the,
I found my way.
As the thoracic surgeon,
They're
your
not-
father used to say, you had aspirations. Give me a drum roll, for God's sake.
This is a really nice. These are really nice photographs.
I'm glad
Yeah,
you
they're
are.
beautiful.
They're gorgeous.
And a lot of them don't have flowers in them at all, so I think
No.
Vincent really should.
I like the one where the room is full of sand. I think that's great.
Well, yeah. Maybe that may be an after photograph.
Nice. Rich, what do you have?
I have a New York Times science piece from Carl Zimmer titled Under the Sea, A Missing Link in the Evolution of Complex Cells. And it describes a paper that was recently published in Nature called Complex Archaea that bridge the gap between prokaryotes and eukaryotes. And basically what it describes is this study, the Nature article, where they looked for archaea-like organisms in sediment from under the Arctic in search of something that might provide the missing link between bacteria or archaea and eukaryotes. And the theory has been for a long time that there must have been some sort of organism that, in my mind, was behaved different than a bacterium because it would have some motility, maybe squishy motility, and a cytoskeleton and some membrane trafficking inside the cell, but still didn't have mitochondria or a nucleus. And that organism would ultimately acquire mitochondria and ultimately differentiate a nucleus. And so these guys think that they found something that fits that because it's clearly related to archaea, but has a lot of eukaryotic stuff suggestive of, they don't have the organism, they got sequence. It has a lot of genes suggestive of stuff that would move membranes around and remodel them and stuff. So I thought that was pretty interesting. And they cite Eugene Koonin in this as being all excited about this being a possible missing link.
Yes.
They mentioned that they got more sediment and the organism is there, but it doesn't live very long. So they don't
Right.
have the conditions right.
Trying to figure out how to grow it, which would be a real challenge for
as-
something that lives 2 miles down in the Arctic.
As you're talking about this, the picture that Vincent has on his screen right now, if you just stop the tardigrade, where the hell did that thing come from? There's nothing else out there like it.
It's a bear.
It's weird.
It's a little bear.
It is weird.
Yeah, well.
It
lots
came
of-
from outer space.
It's okay.
Really, it's a life form unto its own.
Kathy, what do you have?
I picked a site called grantome.com. I can't remember where I found it. I put it in my queue a while back. It's a site that's been launched by data scientists based in Cleveland, Ohio, scientifically trained with PhDs in quantitative disciplines. And so they've done things where they've
Oh, cool.
pulled together all kinds of NIH data. They hope to have DOE and NSF at some point, but at the moment they don't. So that's what you can find out on their About page. Then they have a blog page that has a variety of things on it. I found the home page to be a little bit busy and overwhelming, but if you go to some of those other things, you may come back and find it for things that you're looking for. So those people who really like to obsess about grants, Grantomics, latest tweets about Grantome itself, recently viewed grants, recently added grants, so on. That could be the site for you if that's
This
what
is
you want to
cool.
see.
So I just searched for myself and got a list of all my funding. And I got a graph of my funding over the years, which peaked apparently in 2001.
Which how did you do that?
I just put my name in and searched. Sometimes you have to be brave,
search.
Dickson.
Oh, go where no man has gone
Yeah,
before.
you just put a name in and search. You don't have to ask questions. And it shows me, it's got all of my grant funding on a year-to-year basis. And it's got a graph of it. That's pretty cool. Yeah, I brought in a lot of money to this place.
Wait a
Yeah.
minute. Where'd you put this in the search bar?
Yeah, right in
The
front. You
homepage.
put Condit. And you hit search.
It's not going to be totally accurate for everybody. For example, just before Lois Miller passed away, she made me the the PI on her grants. And not only did I become the PI on her current grant, but I became the PI on all her previous grants. So I'm even listed for her postdoc fellowship, Rich, at ICRF.
Wow.
So there, so if it's based on,
I get all kinds of weird stuff because Condit triggers conditional.
I got mine.
For once, my name works.
I got mine. Wow.
So these folks they're basically the Nate Silvers of grants.
I think so.
They just got all the data. This is really cool.
Then you can click on each one and see the abstract and the funding. Yeah, it's pretty cool. There's a lot of data here. Good stuff.
I'm bookmarking this. This could be very useful.
Hey, look at it. It's got the publications. Dove and Racaniello. Wow. Remember that guy?
Yeah, I do.
It's nice.
And I noticed your grants on the graph. They go up a bit when I joined your lab, and then they went down a bit when
after
I
you
left.
Yeah.
But then they really shot up. So I guess there was some transition.
Here, I could do it with an advance.
Now they're low like I started. So I started, they were low. Now they're back down there.
Wait, what year did TWiV start?
2, 3. So I had 6 apparently grants at one point. Now I have 1. Which year did TWiV start? 2008, I think.
Mm-hmm.
Oh, okay.
There's no bump there. Took a long time.
There's a drop.
Actually, there's a drop, yeah.
Go from 3 to 1.
And you even get the abstract of the grant. Wow.
Yes, you get a lot of, and you get the publications. Dickson,
Yeah,
Okay,
the
well,
digital age is amazing, Dickson.
it's all public information, and these folks have just
That's
pulled it all.
fantastic.
doesn't matter if any of the rest of the TWiV listenership like that you guys did.
Right, that's right. That's right.
This is all
That's pretty
true.
cool. Very nice.
I can get mine if you can go into an advanced search, and there you can put your name in.
See, look, we're all searching for our own support.
Of course.
It's all about us.
Ellen, if you put your name in there, do you get anything?
Oh, I get tons of other stuff. They're
okay.
Simon Dove.
That's very nice.
Probably to do.
All right, my pick has to do with what I've been working on lately. And I've been wrapping up our textbook. I actually finished all the writing, and now I'm wrapping up some figures. So I've been making molecular structures of proteins and assemblies based on X-ray or cryo-EM data. And there are 2 programs I've been using, which I thought I would pass on. Now, there's some of you out there who are listeners are structural biologists, and you will laugh and fall off your chair. Fine. Fine. But for the others, if you ever wanted to make a molecule structure, you can make beautiful ones. There are 2 programs. They're both free. One is called PyMol, which it's a program written in Python, which is the reason why it's there, and molecular. And this is quite nice. And then there's another one called Chimera, which is written by people over at UCSF. It's an NIH-funded project. And both, you basically get a structure file from the protein data bank, and you open it in the program, and you can make all kinds of different images. It's pretty neat. I've been doing this all week, and Dickson has been watching me and asking me what the hell I'm doing.
I have been watching.
And I put
Carefully.
two in the show notes. A beautiful adenovirus fiber and penton base, and the poliovirus RNA polymerase. But if this is a little geeky. I know this won't appeal to everyone, but I think it's pretty cool that you can do this. It's not all that easy, but you can learn it.
Yeah, there's a Mac PyMol version that I learned, and I got help from a structural biologist. Like about an hour training session, and then
that's about what you need. Yeah, so I use the Mac PyMol, and you can, if you have a PC, it'll run on that too. Chimera has versions for different platforms also. I find that PyMol gives you kind of nicer output, but it doesn't always give you the right image from the file, and for some reason, like some molecules you want to show as dimers because that's how they exist in the biological form, and for some reason PyMol doesn't always put both molecules up, whereas Chimera does. So I've been experimenting with both.
Cool.
We have a listener pick from Mauricio. Dear TWiVsters, my twin brother, who is an epidemiologist, sent me this awesome vaccine epidemic game made by the Salathe group. It's extremely fun to play and reinforces the importance of vaccination and quarantine, but in the context of human networks. And it's an online game. It's very cool. Vax.
It is really cool.
Game about epidemic prevention. It's really well done.
Yeah.
Check it out. It's nice. We like it, Mauricio. That will do it for TWiV 336. Our sponsors today were ASM Microbe and ASM Conferences. And you can find all the episodes of TWiV at twiv.tv. Also on iTunes, and of course, any of your podcatcher apps for your mobile device will be able to subscribe to it as well. So do send us your questions and comments. We love to get them. Even if you don't like the show anymore, we still love getting them. And you want to stop listening, that's fine, because you have the right to do as you wish. Send them to TWiV at twiv.tv. Dickson Despommier, thank you for joining us today.
What a pleasure.
You can find Dickson at verticalfarm.com. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Always a good time.
And Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.